Hello students, welcome to the EPG Pathshala. I am Dr. Imtiaz Hassan from Jamia Millia Islamia, a Central University, New Delhi. Today I am going to talk about the module Analytical Ultra-centrifugation from the paper Centrifugation Techniques. So the objective of this module is to provide a detail in the analytical ultracentrifugation, then the instrumentation of the analytical ultracentrifugation, then the method for detection of the molecules, and of course how the data is collected, how the data are analyzed, because these are the important component of the analytical ultracentrifugation. Then another important thing is that how the samples are prepared and its application, that it's used for the different biological purposes, and of course it having the relationship with the many other techniques, so how it is related to the other technique. So the analytical ultracentrifuge is the leading technique for determining the molecular mass, state of dissociation of the solution in the solution, and association constant of the molecules. Even though it is a little used for the supramolecular polymers, this technique has already been shown to represent one of the best option for the characterization of this system. It is used for the biological studies to sediment and separate out the components, and this centrifuge is equipped with the detector system which used to monitor the sample as the sedimentation occurs during the course of centrifugation. Analytical ultracentrifuge is widely used for the determination of sample purity. For example, if you have a sample, you don't know whether it is pure or impure, or of course if it is pure, then at what content it is percent it is pure. So this technique is give you the best idea that how pure is your sample, so you can use for the further study. Then it used for the characterization of assembly and disassembly and mechanism of the biomolecular complex formation. It is also used for the determination of subunit stoichiometry. For example, if you have a big biological complexes like the DNA polymerase or the many proteins that exist in the form of oligomer, so you should know that how many subunits are there, what are the ratio of these subunits as per their size, like the heterodimer, homodimer, and in that term. Then it used to characterize the macromolecular conformational changes, then measure the equilibrium constant that and thermodynamic properties of associating and dissociating system. It is used to characterize the solution state behavior of the molecules under various conditions, and of course it also utilized to measure the thermodynamic properties and as well as the hydrodynamic properties of the molecules under the solution. So this is the widely used technology. Of course it is a very costly technology, but it is widely used for the various biological purposes. So if you talk about its component, it is the analytical centrifuge is actually the preparative ultracentrifuge coupled with the optical system. So it is a simply preparative ultracentrifuge that rotates at a very high speed, and it is coupled with the optical detection system which gives you the dynamic picture of the molecules inside the sample, that is used to measure the sample concentration inside the centrifuge during or after the sedimentation process. So centrifugation parameter and data acquisition under the computer control, so you can see what's going inside the tube before, after, and during the run. An experiment lasting many days performed with the minimal operator intervention. So it is a very long run, so that's why it is coupled with the computer to see the picture from time to time, and then the data are collected and analyzed further for depending upon the application. So here we discuss about the basic component of the analytical ultracentrifuge. You can see here that the analytical ultracentrifuge having a chamber in which the rotors are fixed, and it also equipped with the optical detector system which can provide the actual picture. So the major component are the rotor, which typically spins at 60,000 revolution per minute, while rotor is spinning, 1 gram has apparent weight of 250 kg. And during the rotation, an optical receptor record data from the slide each time it passes. And there is a set of counterweight to keep and ensure the centrifuge is never off balance. So first we discuss about the rotor, that the rotor of an analytical ultracentrifuge must be capable of withstanding enormous gravitational stresses. A typical ultracentrifuge rotor generates a field of 250,000 G at a very high speed of the 60,000 rpm. Under such condition, mass of 1 gram of sample becomes 250 kg. So a rotor must be capable to allow the passage of light through it and the spinning of sample for temperature measurements. The rotor is equipped with 4 holes, which is required for the counterbalance with its reference holes, which provides the calibration of radial distance, leaving 3 positions available for the cells. So this is the schematic diagram of the analytical ultracentrifuge. You can see here a chamber in which the motor is fixed, and at this motor rotor are applied, and this chamber is very sealed, and but it is having the temperature control system, and usually the ultracentrifuge run at a very low temperature so to avoid the heating. And further, this chamber is in the vacuum because at the very high speed the frictional force becomes so high, so the chances of getting fire are very high. That's why the vacuum pumps are connected to this chamber. You can see there is a hole which is used for the detection, and the UV light passes through this and it gives the actual picture to the detector, and that is connected with the computer. In the smaller diagram, you can easily see how the photomultiplier tube can detect what's the phenomena going inside the tube. So this is the basic design of the analytical ultracentrifuge. Cells of the analytical ultracentrifuge are a very important component. It contains sample and a control sector. One contains a solution with the solvent and analyte and to adjust the solvent. So the sector are trapezoidal. The wall emanate the radially from the center to the rotor. This is to ensure that the particles do not hit the sides of the sector and as they move radially away from the center of the rotor. So the cells of analytical ultracentrifuge must also withstand at the stresses caused by the extremely high gravitational field. It is also necessary that the cells must not leak or distort and allow the passage of light through the sample for the measurement of concentration distribution. The sector-shaped sample compartments are essential for sedimenting particles moving along radial lines. The double sector cells held up to take the account for absorbing the component in the solvent to correct the redistribution of solvent component, particularly at high speed. You can see the sedimentation process in the cells is going on, and that simply follows the Archimedes principle that the buoyancy equals to weight of displaced fluid or fluid density into the submerged volume into G. So the forces of equilibrium are the buoyant force and viscous drag equals to the centrifugal force. If you combine all the forces, so the net force will be 0. This is the basic principle for the sedimentation of molecule inside the cell. The buoyant force is equals to mr omega square. Viscous drag is equals to frictional coefficient into the V, and the centrifugal force equals to mr omega square. So the final force, if you combine all these forces, the final net force on a molecule should be 0, and that is the basic principle for the sedimentation of particles inside the centrifugation process. Here we discuss how the solute are distributed in the cell during the centrifugation process. At the time 0, you can see in the first figure that at time t equals to 0, at rest and before the acceleration, all materials are eventually distributed or evenly distributed throughout the cell, and the concentration is constant at radial position. But at particular time like t0 plus n, high speed of rotor. In a sedimentation velocity experiment performed at high rotor speed, the sample will clear in the area around the meniscus and pile up at the bottom of the cell. A moving boundary is formed whose shape is depending upon the concentration, sedimentation and diffusion of the solute. Now we discuss about the optical system that is used for the detection of molecule. So it is based on the absorbance optical system, which measured the sample concentration at a wavelength ranges from 200 to 800 nanometer, and detection of macromolecules containing a strong chromophore. So it can absorb in the visible region, and even the non chromophore containing molecules can be detected at their particular lambda max value. That is based on the Rayleigh interference optical system, that is for the measurement. So there are two systems actually, one is the absorbance optical system, one is the Rayleigh interference optical system used for the measurement of sample concentration based on the refractive index changes. The refractive index changes used for the analysis of macromolecules lacking intense chromophore and a sample that contain a strongly absorbing buffer component like ATP, GTP, DTT, etc. So now we discuss the Schlieren optical system. So in the Schlieren optical system, light is passed through the cell at a specific point. The concentration at that point is changing due to the sedimentation. This result is varying degree of refraction, and this change in radial distance is measured by the displacement of an image on camera, which is then plotted versus time. You can see here in the picture A and picture B. So at initial stage it is having only one peak, but once the centrifugation process starts, it starts moving, and the appearance of the another peak indicating the separation of molecules during the course of time. In the Rayleigh optical system, light is passed through the solvent and sample, and the diffraction pattern created and it is recorded. In order to analyze the data, it is plotted as a refractive index versus the radius of the cell. You expect to see a greater refractive index at far end of the cell due to the sedimentation. You can see here in the graph, and good for the micromolecules that do not absorb in the UV range. So the Schlieren optical system having their own advantage and the Rayleigh optical system having their own advantage. Now you can see the absorption optical system, how it works. It is actually measuring the absorbance versus radius in the cell. The higher the concentration, the stronger the absorbance, and usually nucleic acid absorb at 260 nanometer and protein absorb at 280 nanometer. So it is good for the macromolecules that absorb in the UV range. And you can see here the radius versus absorption is increases with the time, indicating that the separation of molecules occurs. So the equation for change in concentration over time is given expressed by the Lamm equation, that is used to predict the boundary shape and shape that will look like. The given Lamm equation, you can see here that the delta C by delta T equals to 1 by R d by dr, and omega square R square SC minus dr delta C by dr. So this Lamm equation is utilized for the change of concentration over change in the time. Sedimentation constant is another parameter which is measured or which is applied during the analytical ultracentrifugation process. As you are aware that there are two types of forces acting on the particle during the centrifugation. One is the buoyant forces, which is given as m omega square r, where m is the weight of the fluid displaced by the particle. Then the other force is the frictional force, it is given as Fv, so that the net force on the particle is 0, is given as the F net is given as the m omega square r plus Fu. So the sedimentation constant, the net force on the particle is equal to the mass time the acceleration, which is the case the acceleration of the angular velocity squared time of the radius. Here I'll give you an example that if you have a heterogeneous solution, how to analyze using the ultracentrifuge. So what happens if the solution has multiple macromolecules in it? If the solution is spun slowly, then the smallest molecule will not sediment out quickly and will not produce any useful gradient. If the solution is spun very fast, the largest molecule sediment out too fast and not be able to observe. To account for this, the speed is ratcheted up, and after a set of time, in order to able to observe the peak of each macromolecule and type of the solution. So you should simply think that if a tube containing a mixture solution, so during the course of time each component will settle at the different speed, or you can see at the different time. So their settling time can be observed in terms of peak via the detection system, and it will give you an exact idea that what are the molecular mass of the each molecule at the different interval of the time. So as we derive this equation further, we can see that this depends upon the partial specific volume and many other parameters. If we combine all the equation together, then the net force equation is given as m omega square r minus mv naught p rho omega square r equals to m omega square r 1 minus v rho naught. We then place all the variables that are experimentally known on the left side of the equation, and we define as that sedimentation coefficient. So the sedimentation coefficient is given as u by omega s square r, and the sedimentation coefficient is in second and usually around 10 to the power minus 13. So 1s equals to 10 to the power minus 13, and that is also known as 1 Severberg unit. Now we discuss about the data analysis, that the velocity of the concentration boundary is the change in the radial distance over the change in the time, and this velocity is related to the sedimentation constant from our previous definition, that u equals to t rb by dt equals to s omega square r. We can then rearrange this formula and take the integral at the both sides like this. So using this formula we can solve for the sedimentation constant by plotting the time versus the log of radial distance from the concentration boundary. The slope of the plot is omega square, and since omega is known, we can find the sedimentation coefficient from the plot, as you can see in the figure. Sedimentation velocity based method is used for the calculation of the biomolecular shape. Like if in a mixture you have different molecules with a different shape that can easily be determined or can easily be analyzed using analytical ultracentrifuge, and the method is based on the sedimentation velocity. Similarly, the biomolecular conformational changes, for example, if you take a protein and you want to see that it is denaturing during the course of time or denaturing during the addition of certain salt or anything, so you can take a dynamic picture that how the molecules got denatured, because during the denaturation the conformational changes occur and the size got changes. Similarly, if you take an enzyme, it binds to the substrate, then you can find that the binding of substrate induces some conformational changes that can easily be monitored by the sedimentation velocity. So the binding or conformational change based analysis having a wide application in the biological sciences, like the enzyme assembly, binding of DNA to protein, protein to DNA, and many protein and ligands. Then it can be utilized for the assembly and disassembly of biological macromolecular complexes. For example, complex is formed in a certain condition, then you can easily analyze that what are the conditions capable to forming such kind of complex, then how big is complex, what are the component of complex, what are the monomers, what are the dimers, what are the basic component of the monomers of that complex can be analyzed. In the same way, if you see that how these monomers assemble together to form a big complexes, what are the stoichiometry, what are the ratio of the molecule that could bind together to form a complex. Then it also give you the idea about the equilibrium constant for self-associating and dissociating system. So these are the very wide application and most useful application on the biological science. The other part is the sedimentation equilibrium, so sedimentation velocity, and the next is the sedimentation equilibrium. So in the sedimentation equilibrium, it also used for the measurement of molecular mass and subunit stoichiometry. It is useful for the measurement of equilibrium constant for hetero-associating system, like the how the two systems are capable to associate together, what are the binding constant, what are the equilibrium constant, in what condition these equilibrium is changing, and in this equilibrium is shifted towards aggregation side towards the monomer side. For example, a protein is forming oligomers or it is forming aggregates in a certain condition, then what are the factors, how the formation, what are the kinetics of aggregate formation. In the same time, if you want to dissociate an aggregate, so you can also see the what are the kinetics of dissociation of that aggregate. So the equilibrium constant for the hetero associating systems can be measured using the sedimentation equilibrium. In the same time, the equilibrium constant for the association system is measured. So these are the very wide application of the analytical ultracentrifuge. Now we discuss about the standardization method for the sedimentation coefficient, that the S20W. It is given as the equation you can see here, and for the biological macromolecules, the sedimentation constant are usually taken in solution of varying density and viscosity. So the sedimentation constant is adjusted to the standard 20 degrees Celsius in water, and that's why the S20W is taken for consideration, or the standardized sedimentation coefficient for the calculation of the biophysical properties of the molecules. You can see here a typical example of the calculation of sedimentation coefficient, in which the radius is plotted versus the absorbance, and you can see the sedimentation coefficient of a single ideal solute can be determined by measuring the time and position of the midpoint of the boundary using these equation. In the sedimentation velocity experiment, we again plot the radius in centimeter versus the absorbance, and you can see here at initial the meniscus of the solvent, and in the middle part sedimentation occurs. So we usually took the two cells, when one cell blank is there and in other cell the protein solution is there, in which at the top you can see the meniscus, at the lowest part protein, in between boundary and depletion you can see that is against the gravity. So that is utilized for the calculation of the sedimentation velocity experiment for the measurement of S units. So the sedimentation velocity experiment is actually used for the calculation of hydrodynamic information and experimentally determined parameters such as sedimentation coefficient, diffusion constant, frictional factor, molecular mass, and estimation of molecular shape in a solution can be calculated. For these, high rotor speed are utilized and the sedimentation dominates over the diffusion. So these are the parameters can be measured using the sedimentation value experiments using analytical ultracentrifugation techniques. Sedimentation equilibrium experiments are used for the measurement of thermodynamic properties of biological molecules, and it actually determines the parameters such as molecular mass, solution assembly state like the monomer, dimer or oligomer formation. It is used for the measurement of thermodynamic properties such as equilibrium constant, calculation for free energy of the dissociation reaction, and in the same time association also, and utilized for the calculation of thermodynamic parameters. So this technique is based on the mainly on the thermodynamic properties of the molecule, as the sedimentation velocity experiment for the hydrodynamic properties of the molecule. So we discuss about the data analysis in the sedimentation equilibrium. So the graphical data analysis method is actually plot of log concentration versus the R square, and we get the straight line with the slope that is proportional to the m. So it is given by this equation, you can see here in the equation, and it is alternative for more complex system, direct fitting of the sedimentation equilibrium and concentration gradient to the mathematical function used for the calculation of molecular mass. Now we can see the effect of diffusional boundary spreading on the calculation of sedimentation coefficient. The S value of the non-midpoint boundary position from different scans are different because diffusion contributes to the transport of those boundary regions. That's why you can see here in the scan number and versus S value at the above midpoint, at midpoint and below midpoint, there are chances of diffusion, so you can get the different kind of curve. So these are the properties, like the diffusion boundary spreading can be considered for the calculation of sedimentation coefficient, and that sedimentation coefficient is utilized for the calculation of molecular mass of a biological molecules. At last, we summarize that in analytical ultracentrifugation, analytes are separated on the basis of their sedimentation coefficient when they experience a centrifugal force. So this force actually made a molecule to get settled at the different time, and it is usually carried out at a very high speed, less than 60,000 rpm, that is, or even at the more than 1 lakh rpm, depending on what kind of mixture you want to analyze. Then analytical ultracentrifuge can assess complex of size, degree of self-association, as well as giving the measure of the monodispersity in vitro and in vivo system. Then sedimentation equilibrium is an equilibrium established between the sedimentation away from the center and rotation of the diffusion towards the center of the rotation. So this is the all about the analytical ultracentrifuge. I think now you are much aware with this thing. You already seen all detail of the how, what are the structure, what are the components, how it operates, everything. At last, I must say thanks to all of you.